Hello, is anyone there?
Yeah, we're here.
Oh, hi.
Just a minute or two, so just give us a minute or two. So
Sure, no problem.
okay, I think we are 10 now, so we can start. Others will join as we move ahead. Yes, so we know how to do it by now. Okay, can everybody open their video as well so we can see each other? I think my video.
Hi, good to see you.
Yeah, nice to see you. Yeah, everybody can try to open their video so we see our faces. Okay, so we know how we do it now. I'm going to introduce the speaker once again. You should have known that at least to a very good extent by now. But okay, Sarah Carl is originally from the US and studied biology at the University of Chicago. She then moved to the UK where she did a PhD in genomics at the University of Cambridge during her PhD. She started learning bioinformatics in order to analyze her own data and decided to continue in the bioinformatics field. Sarah moved to Switzerland in 2015 to do a postdoc in computational biology. In 2018, she joined the startup Skillite as a data scientist with the goal to discover biomarkers for disease in single-cell data. She is now leading the data science group at Skillite. So join me, everybody, as we welcome Sarah to discuss with us about her career in bioinformatics, and very interesting things about herself, probably a little bit about her research as well. And then towards the end of our session, you can start bringing in your questions. Yeah, Sarah, over to you.
Thank you. Yeah, I'm happy to be here. And I also would encourage anyone to ask questions along the way if you have something that comes up. It's no problem. Yeah, so I can talk a little bit about my career, how I got to this point, and my background in research. As I said, I started off studying genetics, and I was working in the lab, actually in the wet lab, doing experiments and generating some genomics data. And I realized that it would be useful to try and learn how to analyze that data myself instead of having to depend on somebody else. So that's how I first started learning bioinformatics. I was lucky that I had other lab mates who could help me out, and my supervisor at the time was very supportive of that as well. So I did some courses, both online and in person, and that was really how I got started. But it's interesting, I think this field has evolved to some extent since then. So when I was starting, I think everybody was kind of teaching themselves and no one really knew so much what we were doing in a way. And now there's really more and more structured programs, internships like this one, which I think is great, as well as more online courses and just a lot of resources out there to help you get started in bioinformatics if you're interested in it. So I see that as a really positive thing. Let's see, so I worked mainly, so my PhD was working with Drosophila, so fruit flies. And I was mainly looking at epigenetic type of data, so transcription factor, binding, gene regulation. I was doing some ChIP-seq, if you guys are familiar with that technique. Actually a bit of microarrays as well back in the day before the sequencing was fully set up. And then that led to also analyzing some RNA sequencing, so transcriptomics data. And I first started to learn using Perl, which is now a bit outdated maybe as a bioinformatics language, but it was still a good starting point. And then I moved to R mainly. So most of my work I've done in R, but I've also more recently been using Python. And Python I found is really great, especially for machine learning approaches. But yeah, getting a little bit ahead. So when I finished my PhD, I wasn't really sure what I wanted to do. And I already thought that I might not want to stay in academia forever. So I was interested always in sort of biotech and in the startup world. And I actually applied for some positions already when I finished my PhD, but I didn't get any offers. So I got a lot of feedback like, yeah, maybe if you get some more experience first, then you might be more interesting. So I decided to try and do a postdoc in bioinformatics specifically so that I could get more experience with different types of data, different types of analyses, and kind of broaden my expertise. And I was lucky, so I found a nice position at an institute here in Basel, Switzerland, where I live now. And when I was there, I got to work with 2 different labs primarily, but actually also helping sometimes people from different labs around Switzerland. So I really did get exposed to a lot of different data types from a lot of different organisms as well. We worked with C. elegans, worms, mouse, cell culture, yeah, basically most of the different types of models, and still mainly focusing on genomics data of different types. And then I started to get interested in this machine learning thing that was becoming more popular a few years ago. People were talking about this neural networks and deep learning, if you guys have heard about that. And so I started to also do some online courses to learn some more about that. And that was really interesting. So in the end, as I was finishing my time as my postdoc, I started again to look for jobs. And I got contacted by the CEO of this startup, which was really small at the time. There were only 4 people, but they were looking for a data scientist who had some experience with machine learning and with biological data types. And the idea was that they wanted to use this new type of data, new type of biological data from single cells that's becoming more and more widespread. And that could be either protein measurements using flow cytometry or mass cytometry, or it could be single cell RNA sequencing, and really look on individual cell level to try and see which cells and cell states would be predictive of a disease. So that's what we mean when we talk about discovering a biomarker. So it's trying to find a combination of a molecular profile and a cell type that would be indicative of somebody having a particular disease, hopefully more precise than other classical diagnostic methods, and also hopefully earlier. If you think about cancer, for example, well, it's one of the biggest problems in cancer diagnostics is early detection. And so now one of the big trends, or what people are working on, some people are working on trying to detect cancer in circulating tumor cells, so cancer cells that would be going through the bloodstream. Some people are working on trying to detect it even DNA, cell-free DNA, so just DNA that's floating on its own in the bloodstream. And so we really take the cell approach, but we said, okay, let's try and see, first of all, if there are specific changes in cells in the immune system that you can detect early, or in other types of cells that would really indicate the presence of a particular disease and improve this diagnosis. So that's been exciting, and basically it's kind of a wild ride being part of a startup, because you never know if you're going to raise money to be able to keep going or not. Everything is a bit high stakes, but it's also fun because you get to be involved in many different things. Even though I'm the head of the data science team now, which we've grown to 4 people in the team, I'm also involved in helping to, form partnerships or come up with the strategy for the company. I was involved in helping to write a patent for one of the discoveries that we made. So that's something that I find pretty exciting about being in the biotech and the startup space. Yeah, I would open already for some questions if anybody has anything. That was kind of the quick overview of my career path so far. You guys can also write in the chat if you feel like it, if that's easier. I can't hear you, Ole. Are you saying something? You're muted. I think you're still muted. Still don't hear anything.
Sorry, I'm mute.
Oh, there
sorry.
we go.
Yeah,
Okay,
I was
okay.
mute, yeah.
There we go.
Sorry. I said they can use the chat, the group chat, as well as the raise hand, so I can give them access.
Yeah,
okay.
That's great.
Okay, a question just popped in. Thank you, Sarah, for the good talk. I have done some analysis in R, but I always get problems with some packages being incompatible with some R versions and others being compatible. How do you deal with this?
Yeah, that's a common problem, I would say, and that can be really frustrating. But I mean, the best way to deal with it, I think, is by being really on top of kind of keeping your R version up to date and trying to keep packages up to date as well. And I'm not always very good at that. I just recently updated my own R installation and started reinstalling some packages. But yeah, I mean, also, older packages might eventually not work anymore with newer versions, which can also be frustrating. So I usually find it's best to maybe stay with not the very most recent version, but the one right before. That tends to be stable. Yeah. So. Shall I go on to the next question? As a novice, how to explore genomics and cancer genomics? Yeah, broad question. But there's a lot of data that's publicly available now, so that's a good way to get started, I would say. There's the Cancer Genome Atlas, the TGCA, which is, really genetic data or genomic data. But there's also, if you're interested in looking at transcriptomics, there's probably, I'm sure there's a lot of publicly available data on Geo that you can search for by different terms. I would start By looking for interesting papers. I think that's often a really good way to go, is by finding scientific papers that address an interesting question and actually really trying to go through their methods and their analysis and see if you can understand and if you can even repeat it with their data, if their data is available. And even some papers nowadays, it's probably not the majority, but some of them even really publish the code or give a link to, for example, a GitHub repository where you can go and try and reproduce that. So that could be a good way to start getting into it.
Okay, yeah, thank you. Okay, Steven said thanks. Okay, Winfred is asking another question. A quick one. What influenced the choice of your PhD and choice of your career path?
So my PhD, I would say I was really still motivated just by scientific curiosity, I think. And I was lucky that I had the chance to join a group that was doing what I thought was some interesting research and with a supportive supervisor. So in that case, yeah, the program I did, we had like a first year with different rotations. So we got to do a small project in 3 different labs, and then we could choose one to stay in for our PhD. So I knew I wanted to work on developmental biology. That was the other aspect. So, and I was interested in learning more about genomics, which was a pretty new thing, at least to me at the time. So those together made me choose that lab. But I would also say that the overall lab environment was really important. And I would recommend for anybody who's considering to join a lab or a company or any working environment that it's super important to make sure that like the people you're working with are people that you enjoy working with and that you feel supported. And I mean, like science is hard. Things often go wrong, no matter what. So it's always better if the people around you are creating a positive environment and helping you rather than, you know, being competitive or trying to put blame or something like that. I think that's really important for a PhD.
Okay, there is no question yet. Maybe I can ask mine.
Sure.
Yeah, I wanted to ask, what do you think between science in the academia and science as in the, let's say, startup companies? Which one do you like most and why?
They're different for sure, and I like both. So, okay, science in academia is, I mean, if you really are interested in like more fundamental science or basic science questions, I mean, academia is the best place to do it, because if you have funding, then you have basically free reign to pursue whatever question is interesting to you. Obviously, when you work in a company, I mean, usually at least the goal is to make money as well. So you have to, in the end, have something that you can sell. And I think that's okay, but it does constrain sometimes the topics that you want to work on. So like in my case, I find it super interesting what we're doing. But sometimes I have to say, okay, actually, no, we can't spend the time to really dig in detail in this particular data set because it's not really relevant to the product that we're trying to make. So we should rather refocus on some other topic that's that fits better in a particular, you know, the direction that we decided to go in as a company. But yeah, then I think, so academic science can, when it's working well, it can be, you can have really great collaborations as well, which is nice. When it's not working well, then yeah, you can have a lot of competition and sometimes bad behavior. So that's another thing. I think I'm very much in favor of like open science and open access and sharing data and sharing protocols as much as possible. I think this is becoming more and more widespread, and more people are recognizing the importance. Yeah, you should also watch out maybe for people who want to just kind of hoard all their data for themselves and not share it with others. Yeah. This can be really frustrating to work with.
Okay, I think we have some questions. How can we bring RNA analysis into forensic science?
Oh, interesting. I have to say I'm not an expert on forensic science. I don't really know that much about how it's done right now. I mean, RNA is a little bit challenging just as a molecule to work with because it's a lot less stable than DNA. So if I think about like trying to find traces of molecules that are left in a particular site, then you're much more likely to be able to find DNA than RNA. Yeah, that's interesting. I don't know. I'm sorry. I think I would have to think about it some more, what the possible
Yeah.
applications would be.
Well, I think forensics, it's more of DNA.
Mm-hmm. I think so. Also, when you think like RNA, what's powerful about using RNA is that you capture like the current state of the system,
Yeah,
right?
exactly. Yeah.
But this is not always what you want. Sometimes you don't want to find things that are changing. Sometimes you want just like stable baseline that you can always trace back. And so DNA would be much better for that.
Okay. I think the person who asked that must have heard. Carol Villas Bohas is asking, what made you being noticed by the startup?
So I always say, I kind of hate this, but it's true that networks are really important for getting jobs and this kind of thing. And in this case, there was a big network aspect. So the founder of this startup had done his PhD in the same institution where I was working. And when they were looking for a data scientist, he contacted my boss at the time, who was the head of the computational biology group there, and asked if he knew of anybody who might be interested. And then he kind of passed that job description on to me. But so, I mean, that being said, you know, it's always good to build strong relationships with people around you, and you know, networking not in the superficial sense of just like making connections on LinkedIn, but you know, like what we're doing now in this internship as well is a great example. Like when you work together with people and you can show them that you bring something to the table and that you're trustworthy, I think that goes a long way. And then in terms of actual skills, I think, I think there's a big niche right now for people who have some experience generally with data analysis and either statistics or machine learning. This is really, I mean, this is getting applied in more and more fields, but I think it's also really valuable to have a domain-specific knowledge that comes along with that. So that could be biology, genomics, cancer biology. It could be drug discovery, or you know, vaccine development, anything, whatever you're interested in. But if you can combine that really specific knowledge that not everybody has with a more general understanding of data analysis methods, I think that makes you a very attractive candidate to a lot of companies right now.
Thank you very much once again. So yeah, some very huge questions here. Just before we go to the huge question, I have one here. Thank you for the insightful talk. Are you hoping to mentee? I'm using Drosophila model for my PhD research, and I'm relatively new to genomics. From Adipo Joyodich.
So I don't know exactly what you're asking for here. I'm open to, I mean, you can send me a message on the Slack channel if you have some specific questions. I'm happy to try and help. I can't, you know, supervise somebody fully, you know, for research at this point. I don't have the time, but in a more informal way, I'm happy to try and answer questions or give some advice on the science.
Yeah, I think that's what the person means. Of course, he or she should have a supervisor by now.
Yeah, I think so. Yeah, if you're already starting with your PhD research, then sure.
Maruf Ahmed Boulian is asking, okay, thank you, Sarah, for orienting us with your work. I'm a physician and I was fascinated with programming from childhood, so that led me to explore bioinformatics as a career path. I'm currently working on machine learning on R/Python. and genomics data in bioconductor in R. Should I also learn BioPython? I'm currently dipping my toes into single-cell RNA-seq data too. What are the benefits of single-cell analysis in your opinion, and what insights does it bring to the table?
Yeah, lots of questions. So BioPython, I don't see it used very often in my field. There could be fields in which it's more commonly used, but for genomics, what I see more often is similar to what you described there. So doing the more genomic side with bioconductor and R, and the more machine learning side with Python. I would recommend looking at, for example, scikit-learn is a Python library that's specialized for machine learning. Yeah, I think the combination of these two and plotting, the data visualization is also often really nice in R, although of course there are also Python libraries for that. But yeah, that's probably what I see as the most common combination. About single-cell RNA-seq data, so I think it's the benefit, well, at least on a theoretical level, is that you get a higher resolution in the results, right? So like in our company, we use this analogy sometimes when we're explaining to people who aren't biologists of a fruit salad. So if you imagine you're eating a fruit salad and there's something sour in it that doesn't taste good and you want to find out what's wrong. So the classical bulk analysis would be that you take all the fruit and you blend it into a smoothie, and then you try and figure out from your smoothie what's bad. But that's really difficult because you've averaged now across all of the fruits that you started with, right? Whereas a single cell analysis allows you to really pick out every piece of fruit individually, and finally you might be like, oh, okay, here's this banana that was rotten or something, and that's what makes my salad taste bad. So yeah, the equivalent of that obviously in biology is that you can look at different cell types individually instead of averaging across an entire tissue, and you get a higher resolution that way. But there are difficulties too. Of course, it's more expensive to perform. Because you need not only specific equipment to be able to isolate the cells, but you also need to sequence more usually than if you're doing bulk sequencing. And the analysis is not always so straightforward, although there are more and more standardized pipelines and libraries that are being used today. In R in particular, there's a library called Surat, which you may already be familiar with if you're looking at single-cell RNA-seq. And in Python, I think ScanPy is the most commonly used one. So both of those have pretty robust pipelines that can be used for single-cell analysis.
Thank you very much. I think I also tried to come into it, but it's a little bit difficult for me right now, and my program is currently disturbing me. Okay, you answered someone's question and saying, great, Sarah. Thanks for the answers and congratulations on your achievements.
Thank you.
Ayodiji is also saying thanks. We've just used 30 minutes and I don't think I'm getting more questions. I don't know why. Okay. Oh, sorry. Okay. Is anybody still asking questions? So anybody wants to talk and maybe talk to Sarah?
Quiet. That's okay.
Don't be shy.
Well, I hope everyone found this interesting and that you're finding the internship useful as well. I think it's a really cool program and idea.
Yeah, some of them said they are scared when I send my messages.
Yeah, I've seen that too. Oh, come on. It's It's not that bad. No, it's a little tough because people have to figure things out a lot by themselves. But I think that's how it works in the real world as well.
Yeah, exactly.
So,
we're trying to model the real world, so
yeah, exactly. Another general tip I would give for people who are interested in learning, any kind of analysis or new languages, is to try and have a real data set that you're working on when you do it, because I think you always, it's more motivating, first of all, if you can actually answer a question or solve a problem. And I think you remember it better, or you learn it better as well, if you're working on a real world situation instead of just picking something randomly from a tutorial or whatever. So I think that's, if you have the chance to analyze your own data, then that's great. Or if you find some public data, then it's always better to have a real question. Oh, looks like there's one more question that came up.
okay. Yeah. I think some questions came up. Okay, somebody said for TSNE plot generation, I can't recall the full meaning of TSNE right now. It is advised to reduce the dimensionality of the data. Okay, to reduce the dimensionality if the data is very high dimensional, like with PC. So my question, what's the cutoff for reducing data before doing a TSNE plot?
So that's a bit more specific technical question, but okay, I think I'm not totally sure if this person means what's the cutoff that tells you should reduce the dimensionality, or how many dimensions you should reduce it to. But I'll try to answer both. So the main reason, honestly, to reduce the dimensionality before generating a t-SNE plot, that's usually how we pronounce it, t-SNE, is to speed things up, because t-SNE can take a really, really long time if you're working with very high dimensional data sets. So if you have more than 100 or so dimensions, I would say it's definitely recommended to reduce it, just so that you don't end up waiting days for your t-SNE plot to run. The question of how many dimensions to keep. So if you do a PCA dimension reduction, this is a tricky one, and there's no hard and fast answer. But there are some plots that you can make, for example, that will let you look at what percentage of variation in your data is captured by each component that is resulting from the PCA. And so usually this will start off high, because that's the whole point of PCA, is that it tries to capture the most variation in the first component and then decreasing. And so sometimes you can find kind of an elbow plot where at a certain point there's an inflection in the curve, and you don't really capture that much more variation anymore. So that's kind of a heuristic to see where to set this cutoff. I think that's a reasonable approach.
Exactly. You're right. I think you have some, I don't know whether it's to the way you were teaching just now. I think you have some flair for teaching as well.
Oh, thank you. That's nice. It's fun to teach, especially when students are motivated and interested.
Okay. I think, yes, and it's just, okay. So, he said, thanks a lot, Sarah.
You're welcome.
Maruf Hamad. Still waiting for more questions, please. More questions should come in.
So maybe I can ask a question for people here.
Okay,
So how many people here are already doing some kind of bioinformatics in their everyday work, or how many people are just interested in trying to start? And if you are doing bioinformatics, what kind of data or what kind of organisms are you working with?
You can raise your hand.
In the chat.
Yeah, or probably the chat. I think Maruf has already started working.
Yeah,
Okay.
it sounds like it.
Someone said
Someone to me.
I'm a total newbie.
That's okay. You have to start somewhere, right?
Yeah, yeah, yeah.
Okay.
I'm
Keep
just
trying to
trying to
start.
start. Okay.
Hmm.
For you to have survived HackBio to this stage, then that means you started something. I mean, you've learned a good deal.
Hmm.
Okay.
Interested? Okay, working on RNA-seq, new to
Continue
bioinformatics.
yet, but highly interested in making a career out of it.
Hmm.
Shifumi, I'm new to bioinformatics, Lale, but so far it is interesting. Okay.
Hmm. Someone says it seems scary. What seems scary about it? It's not that scary. Oh, wow, okay. Someone's working on designing a variant calling pipeline.
Okay,
That sounds
genome.
pretty advanced.
I'm just starting. I have my undergraduate in biochemistry and presently I'm running my MSc in genetics. Okay.
Well, for people who are just starting, I mean, don't get discouraged. I think it's a great time to get into the field, because it's just, I mean, there's so many possibilities. There's so much data that's out there, and also code that you can use and collaborate on. It can seem maybe overwhelming at first, but it's a good problem to have, that there's a lot of stuff that's out there rather than not being able to find anything. And especially nowadays that you have an internet connection anywhere in the world, you can access this stuff. It doesn't matter so much where you're located. I think that's a great thing about the field.
Someone is asking again, are you open to mentorship?
Well, like I said before, I'm open to mentoring in a more informal way. But if you have some questions, you can just write me on the Slack. It's fine.
You can, I don't know if some people do not know how to do that on Slack. You just try to search. I mean, probably I can do a small demo just to, my workspace is always very heavy with so many questions.
Everyone wants to talk to you.
Yeah, you can just come to the search panel and then Sarah. I think there are two Sarah.
I'm the Sarah Carl. That's me.
Yeah.
No.
So here you can. I don't want them to see our charts. Okay. Okay, someone said working with genomics, but I am still exploring what I like most and what each field.
Great. Yeah, that's good.
Okay, Tiffany says, I just started learning bioinformatics, and with the help of this internship, I started learning by Python and R. Yes, I wanted to ask, what do you say to women in science? Generally?
What do I say to remain in science?
Yeah, like your opinion about women contribution and participation in science.
Oh, women in science. Sorry, I didn't get it at first. Oh yeah.
Don't mind my accent.
No, I'm sorry. I think it's really great to see how well. First of all, I think it's great how you've encouraged women in science through this internship. In my experience in biology, women are becoming more and more common, let's say, or like more and more women are going into science. Also in bioinformatics, I would say. But what we see often is there's still like at the lower levels, let's say, like starting to study, then it's maybe 50-50, but then you still have less women as you go more senior. So among professors, it's still dominated by men. So I think, this will take time to change, but I think it's really important that women who are motivated, keep working towards trying to bring this diversity and gender equality. And I mean, yeah, there's also, I think it will be somewhat different in every place depending on where you live, but it's also probably common around the world that women have often different responsibilities or different expectations. Sometimes they have more childcare responsibilities than men, things like that. And this can be difficult, or sometimes it can be discriminated against by employers. So I think it's important to try and change that as well, to give more flexibility. I mean, this is a big topic that I've seen come up with the coronavirus pandemic, that a lot of women academics have had, have found it really difficult to get any work done because they had to stay at home and take care of their kids, whereas the men said, oh, it's great, I don't have anything to do all day, I can just work on my research. It's completely unbalanced, but yeah, I mean, it can only change when we talk more about it and are kind of open to working together and helping each other. So, but I think it does change, and I think if I look back at the previous generation, they faced maybe more open sexism and more open discrimination that I don't see myself. I've had a number of male mentors who've really helped support me and support my career. I appreciate that. So I never felt that I was, discouraged or told that I couldn't do science because I was a woman. So anything is the opposite. So.
Okay, thank you very much. Thank you very much. I think that should encourage the women and the ladies on this call. I believe question just came in. Okay, Sarah, my question goes like this. When I see a lot of overwhelming information, especially once what happened to my chat? Okay. I think the questions just disappeared. Did Tiffany just send empty spaces?
Oh no, it's clapping hands. I can see it. It's a emoji. Thanks.
When I see a lot of overwhelming information, especially once I don't understand, I begin to feel discouraged. How do I deal with that?
Yeah, that's understandable. Sometimes I do too. I think the best advice I have is to try and start small and specific. So not try to read and understand everything at once, but try to break it down and just pick one thing, one piece of code or one part of a paper, and really try and dig in and see if you can understand that, and then gradually expand from there. But also you have this community here, so don't feel afraid to reach out to ask others. I know there are other experts in different fields taking part in the internship, so use each other's resources and knowledge as well.
Okay, Maruf is asking again. So this is not strictly related to genomics or transcriptomics, but I would like Sarah to go through a manuscript writing process, if possible. I have written 6 to 7 manuscripts so far and constantly exploring more efficient ways to do it. If possible, it would be very helpful if you could shed some light on your manuscript writing process, since we're also supposed to write a manuscript in a relatively short time. Yeah,
that's true. Yeah, interesting topic. I'm also maybe not the world expert on this, but I can give you a few insights. So I think it can often be helpful to actually start with the figures. That's what I've done in a lot of collaborations I've been part of, because when you have to make figures, then you're forced to think about what are really the important results that you've found. And then you also have to kind of structure a little bit how you would present those results. So which things go together, which things don't. Then what I like to do, what I find usually helpful, is to write the methods section, because if you've been doing the analysis, then you should know what methods you used. So that's usually pretty easy to do. You just have to kind of go back over your code and think about every step that you did. So then if you have the methods and the figures, it should be relatively easy to write the results, because you just have to think about what you show in each of your figures and kind of how you got there. And then, the trickiest part is probably always the discussion, because that's where you have to kind of give a bigger picture and think about what's important. But then you want to bring in probably literature, background, all this kind of thing. But usually that's the order I write in, and sometimes I even write the introduction last, because once you've already written the rest of the paper, then you can maybe think more clearly actually about how to introduce it and how to catch people's attention at the beginning.
Okay. Yeah. Thank you. I think that I also follow. I use the images myself, the pictures to describe, and then from there I can build up on top. Okay. Shifumi is asking for starters in bioinformatics. Where do you advise we start from? I think this question comes up a lot of times. What
Yeah.
languages should we learn first that are still relevant in the field? What courses can we take?
So, okay, the languages, I would say the two most relevant or most commonly used ones are R and Python. And you've probably seen this coming up again and again. It's also always useful to have a bit of experience working in Bash or just on the command line, because you'll always need to do that. That being said, I would probably pick either R or Python to start with, because they are fairly different. And if you try to learn both at the same time, I think it would be confusing. And it probably doesn't matter that much which one you pick. Each one has its own strengths and weaknesses, but I usually tell people that what matters more is actually the community that's using that language. So what packages are being developed, how other people are applying them. Because if you want to solve a particular problem, then it's better if you have a lot of tools and a lot of other people available to try and help you with that problem. So I would try to start from kind of the type of problem you want to be able to solve, and then look for what other people are doing and what's available. Courses, so it's been a while since I've looked, but I'm pretty sure there are some bioinformatics courses on Coursera, for example, that might be useful. I put a link a while ago in the resources channel to a platform called Rosalind, and I think that's a nice intro to both programming and bioinformatics, because it gives you a series of exercises, basically starting from easy and getting harder and harder, that introduce different concepts and kind of bioinformatics algorithms and things like that. So you can kind of learn those concepts at the same time as you improve your programming skills. So that's something you can work through on your own.
I recall the Rosalind link. Maybe I'll try to locate it and share it once again.
Okay, sure.
Okay.
Oh yeah, okay. Somebody else shared already. Thanks.
Thank you, Olutunde. Right? Yeah, thank you. Any more questions? I don't know. Are we getting any questions? Okay, she said, thank you so much, Sarah.
You're welcome. Okay, more questions? More questions? Okay, another person said thank you. Okay, maybe one more last question from me. So that people won't think it's just overly difficult working in HackBio internship. So can you, for example, if a new task comes up or probably a new research comes up, how do you start from, let's say, ideating, thinking through the solution, and then identifying your softwares and just trying to get things through by yourself? Okay, just an overview. Yeah. Yeah, that's a really important process, let's say, in research, I think. So I like to start from the literature. So looking for papers that are published where people did something similar, and looking on PubMed or or even Google Scholar or something like that, just trying to search in the general topic. Or of course, if you can ask anybody else for a recommendation, if you know somebody in the field, then that it's always, I think it's important to get a sense of the background of the problem, because usually there are, let's say, even if there's a similar approach for analysis, different data sets, different questions have different things you need to consider. So it's important to try and understand like what might be specific to this problem that I need to take into account. And then, then you can look and see what those people use to analyze their data, what tools they used. Try and look a little bit at the documentation for those tools and see if it seems feasible, that you would be able to install it, that you would be able to work with it. And then, just try things out. I mean, you can start with a smaller data set, maybe just kind of an example data set, or a toy data set to get a sense for the tools and to see if you manage to make it work before actually trying on a real data set. But yeah, I would always just recommend starting by doing research in the literature, by trying to understand what's been done before, what are the pitfalls to take into account, what are the possible ways to move forward. It's the same basic process.
Okay, thank you very much. Thank you very much for the session today. So it's supposed to be 1 hour, and I think we have less than 8 minutes right now.
Mm-hmm. Okay. Yeah, I think it.
Yeah, you were about to say something?
No, it was nice to have a chance to talk with everybody, and everyone asked their questions if they had some.
Yeah. Hopefully everybody was able to ask and pour their minds and at least listen to you. You were an inspiration to all of us.
Thank you.
So thank you for honoring the call. Probably we'll still get one more question, but if none, I will say thank you once again. Thank you so far for your support in the internship. I mean, the way you energize and inspire the interns, you provide some resources, you provide guidance, even in the framing of the research topics. You have really, you have really helped us. And somebody said, thank you, Sarah. You have really shared a lot. Okay, thank you.
Well, you guys are all welcome. And I wish everybody best of luck with the internship and also with your careers going forward.
Yeah. Okay, so if there is no more question right now, everybody's just saying thank you. Okay. Thank you once again, and to everybody, thank you for joining the call and for interacting with Sarah. We hope to. So probably everybody should. It's more like the town hall meeting now. Everybody can go back to work.
Right. Yeah, you guys have work to do, so keep it up.
Thank you very much. Have a nice day. Have a nice day.
You too.
So we do something. Everybody unmutes and they say thank you to you. So I've unmuted everybody. So you can say thank you, Sarah.
Thank you.
Oh, thank you very much, Sarah. Thank you, Sarah.
You're welcome.
Thank you.
Thank you, Sarah.
Thank you.
That's really nice.
Okay,
Great.
have a nice day.
Okay, everybody.
Yeah.
Bye.
Bye.